Hello everyone. Today I'm going to share with you an experiment which was done by the NIUMAG NMR for food research. So this is the experiment about the determination of freezing point and thermally induced denaturation temperature of salmon flesh. For the determination of freezing point, we changed the sample temperature from 10 degree to minus 20 degree, and the testing was done every 2 degree. For the determination of the thermally induced denaturation temperature, we changed the sample temperature from 30 degree to 60 degree, and the testing was done every 2 degree. So you can see that because our test temperature range is very large, we can conduct online research on samples of about 200 grams. In fact, the principle of this experiment is that we judge the freezing point or denaturation of salmon flesh by studying the changes in the proportion of water in different states of salmon flesh under the variable temperature conditions. There are 3 states of water: free water, bound water, and confined water. The relaxation time of the 3 types water are different. So here is the result of freezing process of salmon flesh. The left graph shows how T2 changes by the sample temperature. The initial stage, we only have T22, then we got T21, T22, T23. These are the relaxation of different states of water, and we can see all of T2 decrease by the decrease of temperature with nonlinear relationship. This means some change happens. Put the apex at the different temperatures on the right graph. T21, the confined water, basically has no change, and T22, the bound water, moves to the left. T23, free water, changes the most obvious. The temperature decreases and T2 becomes shorter, indicating that it has become bound water. Let's look at the result from the image level. We got a similar freezing temperature by 2 different technologies. The graph C is the result of NMR, and graph D is the result of DSC. DSC is the differential scanning calorimeter. The graph C is the relationship between relaxation time and temperature. The intersection point is obtained by linear fitting, and it is considered that its point has undergone a phase transition, that is the freezing point, and temperature is minus 8.08 degrees, and the DSC proved that minus 8.27 degrees is the freezing point temperature. The more sample we test, we think NMR result is more meaningful. And here, graph E shows the MRI of salmon during the freezing. It can be seen from the graph, it is basically unchanged above 0 degree, and it almost disappears below minus 4 degree, because as the temperature drops, free water freezes. This signal of ice cannot be acquired, so the image gets dark. Now let's look at the process of heating up. We got 2 T2 in the heating process, T21 here and T22 here. And we can see T21 keeps stable. That means the motility of water did not change so much. On the other hand, T22 tells another story. It decreased sharply before 40 degree. I think the environment of water in the sample changes a lot in this process. T22, the free water, it was free water at the beginning. When the temperature rises, part of it evaporates, and the remaining part becomes bound water. No matter how high the temperature rises, it is difficult to evaporate. The bound water T2 warm remains unchanged. So the temperature point of salmon protein denaturation is around 40 degrees. Let's check. The graph C shows T2 verse temperature, and we got heat denaturation temperature by two linear models. The red cycle I marked here tells us the temperature is 42.16 degree. The denaturation temperature tested by the DSC on the graph D is 41.53 degree, which are very close. But from the MRI, there is no significant change, mainly because the relaxation time on the heating process changes little, and the nuclear magnetic field cannot distinguish it. So now let's take a look at this NMI 20 NMR. This is one of the food research NMR from NIUMAG. The previous salmon flesh experiment about the freezing point or the denaturation point is a good explanation of it. The probe of this instrument is optioned, either 40 millimeter or 25 millimeter or 15 millimeter. The customers can test different sized samples. It can also equip the variable temperature equipments. The last technology is from minus 50 degree to 200 degree. And here I listed some applications of this instrument. The first is the quantitative determination of water or oil, and also analysis the comparison of water content in different binding states. Also the spatial distribution of water or oil, and multi-technology combination, on-site test. Okay, so that's all. Thank you. You can follow us and we will share more about the NMR application.